Ani, bonjour, welcome everyone to the fifth and final installment of CROSH COVID Conversations. My name is Toby Mankus, and I'm the Science Communication Officer at CROSH, the Center for Research in Occupational Safety and Health. We're streaming live from the CROSH lab at Laurentian University. We acknowledge the Robinson-Huron Treaty of 1850 and recognize that Laurentian University is located on the traditional grounds of Tikkamishing Anishinaabe. The city of Greater Sudbury also includes the traditional lands of Wahnapitae First Nation. CROSH COVID Conversations is a 5-part webinar series of the intersection of occupational safety and health and COVID-19. Our panelists will speak for 15 minutes each, and then we'll have 10 minutes of audience questions. You can enter the questions at any time in the chat function of the platform you're watching this on. I'll then ask your questions to our panelists during the Q&A section. Today, we'll be discussing biological mechanisms of COVID-19 infection, herd immunity, how vaccines work, and why vaccination is important for the workplace. Our panelists are Dr. Sandra Dorman and Dr. Caitlin Malarkey. Dr. Sandra Dorman is a full professor in the Faculty of Health at Laurentian University and is the director of CROSH. Her expertise is in respiratory physiology and immunology, and her research program focuses on health promotion and disease prevention in the occupational setting. She has a track record for successfully addressing workplace-identified problems and communicating information back to the host organization. She's authored over 25 papers and received over $5 million in research funding. Thank you for joining us today, Dr. Dorman.
Thank you for having me, and welcome everybody for our final webinar of this series. I'm excited to talk to you today about immunology. It's one of my favorite topics, so let's dive in. I wanted to talk to you about coronavirus, also more commonly called COVID-19. And I want to talk to you a little bit about how it actually infects our bodies so that you have a hopefully better understanding of the different ways that scientists and researchers are trying to use therapeutics to prevent infection or to help us get better more quickly. So COVID-19 as a short form comes from coronavirus disease 2019 when it started, right? And I like to say the corona piece because corona means crown, coronaviruses have been around for a long time. They're a recognized group of viruses, and they were called corona because when scientists first put them like on an electron microscope view, you could see the circle of the virus and then all of these studdings that created like a halo around the virus. And whimsically, I guess, the scientist who looked at that said, it looks like the virus is wearing a crown. And they called it corona. That's kind of interesting and kind of pertinent in that, when you look at the actions of the coronavirus, that crown feature is really important for how the virus infects us. And so, I'm sure all of you at this point have seen some kind of cartoon image representation of the coronavirus. And they typically show it, it's a beach ball with these red studs all the way around it. And the red studs are what create that crown effect. And if you zoomed in on any one of those crowns or those red studs, to me, they kind of look like a broccoli stalk or a tree, right? And that stud has a name. They call it the spike protein, which is, I find immunological names are often quite cool like that. So it's the spike protein. And scientists have broken the spike protein down into 2 parts. So the tree part or the bushy part at the top is the spike 1 portion of the protein, very clever, S1, and the stalk part is S2, or the spike protein portion 2. And those 2 pieces are actually both very important in how you become infected with coronavirus. The stalk, you'll notice, is the part that sticks right into the virus, right? And the tree-like portion is what is exposed to the outside world. And so when the virus is kind of inhaled into you and flying into your body, it's the tree-like portion that's going to stick to the outside of your cell. It's going to allow it to make contact with your cell. And actually, it's not just sticking anywhere on the cell, it's sticking on a very specific protein. And that kind of allows it to dock on the outside of the cell. That's the S1 protein, okay? The S2 protein, if you imagine your cells, they're like castles. They keep everything out, but there's portals to the castles. The coronavirus is sticking to one of those portals. The S1 part sticks to the door. The S2 part turns the knob and allows them to go in. So the S2 protein is what actually allows the virus to get inside your cell and infect it, right? So you can see, like, before even going too much farther, why targeting both the S1 and or the S2 protein on coronaviruses would be important to preventing infection, because they're both so critical to how the virus gets into your cells, right? So I want to segue from there and talk about, okay, well, what's the portal on your cell? What is it? What is the virus sticking to? And that is also a protein that's called ACE2 protein, basically. And what's interesting about ACE2 protein, well, first and foremost, where do you find ACE2 proteins? Well, there's lots of ACE2 proteins on your lung cells, lots on your blood vessels, on your gut, and on your kidney, right? So that makes sense, because here's a virus that is predominantly being passed through the airways, right? It wouldn't be successful if it didn't have something to bind onto in the airways. You can also see how it can spread to other sites because there's receptor in the blood vessels. You can also see how actually, why people will often have gastrointestinal symptoms when they're infected with COVID. And how, when you go to the bathroom, then whether you pee or you poo, you're shedding virus into the wastewater as well. Which, if you haven't seen, I think it was webinar number 3 on one of the things we looked at was measuring COVID in wastewater. So you can go back and check that out. Anyways, so ACE2 protein is in a lot of important organs in your body that are related to infection. The other thing about ACE2, most of you are aware that there's a certain health profile that's linked with worser outcomes with COVID-19 disease, right? Age, what your baseline health is, as well as whether you have a healthy diet and regular exercise. And actually the functioning of the ACE2 protein links up to that health profile, so that it may be part of the reason why some people have worse outcomes when they're infected with COVID-19 than others. So I want to talk about that a little bit and explain that. So what does ACE2 protein do? Basically, it balances the levels of 2 important hormones in your body, angiotensin 2 and angiotensin 1-7. I know the names are kind of long, but angiotensin 2 is a hormone that's involved in increasing blood pressure and increasing inflammation in your body. Angiotensin 1-7 is a hormone that's involved in decreasing blood pressure and reducing inflammation. And most of the body things, it's not that one is good or one is bad, right? When stuff happens, sometimes your blood pressure needs to go up, and then your blood pressure needs to go back down. And that's why you have hormones that create a balance like that, right? The ACE2 receptor takes angiotensin 2 and converts it to angiotensin 1-7, right? And what you see is that most people, when they're younger, have higher levels of angiotensin 1-7. Aging causes a switch in that direction, as do certain types of disease. And I want to say one other thing. So because ACE2 protein converts angiotensin 2 into angiotensin 1-7, when it makes that conversion, it actually changes shape a bit. And so the SARS COVID-19 binds to the ACE2 receptor before it converts angiotensin 2. After it converts the hormones, it changes shape, and now SARS can't bind anymore. So you could see how a person with one type of profile where they had high angiotensin 1,7 and a converted ACE2 protein receptor might be better protected than a person who has high angiotensin 2, which is causing high blood pressure and more inflammation, and an open receptor, right? Now, I do want to make clear that that's not to say that ACE2 receptor is the be-all and end-all for a person's susceptibility to COVID disease. Certainly, I mean, first and foremost, you've got, how your viral load upon exposure. The more virus you have is obviously going to make an outcome worse, right? You also have your individual's host immune response, and the more robust your immune response is, the better protected you are too. I'm just saying that scientists also think that this ACE2 protein may have a role and may be contributing to worse outcomes in certain individuals who have that profile. Now, the reason why we care about that again is because it gives us opportunities for therapies, right? How can we treat or prevent COVID-19 infections? And so just understanding those stages of infection now, I think you can see some clear pathways. We can target the S1 protein and prevent the virus from attaching to the cells at all. We could target the S2 protein and prevent the virus from actually entering the cell, right? But we could also look at therapies, and there are drugs that already exist that can either target angiotensin 2 levels, target the configuration around ACE2 proteins, the number of their expression. And I'll just give you an example that I thought was really exciting. If I say it right, there's a pharmaceutical company called Apeiron, and it's actually running clinical trials now where it's putting a floating form, a soluble form of ACE2 protein in patients so that it basically would mop up SARS protein. So remember, normally ACE2 receptor or ACE2 protein is the door that's attached to the cell, right? But this company has taken the door off and just thrown it floating around in your bloodstream. So the SARS can still bind to it, but there's no cell for it to enter. And so it ties up a bunch of SARS viruses, and then they can be mopped up and removed. So, knowing that a vaccine is coming, but it may not be available to everybody, the more options we have for different therapies, I think the more meaningful. The last thing I just wanted to say, there's two actually things I want to say. The first is you may have heard of angiotensin 2 before, and you may in fact be on medications that target either the enzyme that activates angiotensin 2 or that blocks binding of angiotensin 2 to blunt its effect, right? There are common blood pressure medications that people use that target that hormone or its receptors. And so I do want you to know that it's important to continue to take those medications if you get COVID-19. The research has shown that people do better when they continue to take those medications. And in fact, those medications may be offering a protective effect for people in either getting or in not having severe consequences from COVID. So stay on your high blood pressure medication. The other thing I wanted to tell you is Health Canada has created this wonderful video. It's called COVID and the ACE2 surface protein. And it really takes you through a lot more of the biology. And I think it's about a 5-minute video. It's not even that long, but it shows you pictographically what's happening. I recommend that you view it if you want to get a better understanding of like, what does angiotensin 2 do and why does it cause inflammation, that sort of thing. And lastly, I'll just throw out there, because I was involved in the panel a couple of weeks ago on masks, and there was an article that came out in the British Medical Journal just yesterday that looked at all the different fabrics that people have been using for masks. And so if you want to look that up, I recommend it. I didn't have time to read the whole thing in depth, but it looks like they all do a pretty decent job, is what it says. All right, with that, I will hand off to Dr. Malarkey.
Thank you very much for that, Sandra. I really liked how you broke down kind of in very understandable kind of physical terms how that works. And I think we're going to dive some more into that in the question and answer. But first, our next panelist is Dr. Caitlin Malarkey. She's an assistant professor and associate chair of undergraduate education in the Department of Biochemistry and Biomedical Sciences at McMaster University. For many years, her research focused on novel universal influenza vaccine approaches. More recently, though, she's become immersed in the scholarship of teaching and learning with a specific emphasis on exploring advanced methods of delivering learning content. With extensive training and expertise in infectious diseases and vaccine development, Dr. Mullarkey teaches virology, cell biology, biochemistry, immunology, and advanced laboratory techniques to undergraduates at all levels. So thank you very much for being with us today.
Thank you so much for having me. I'm delighted to be able to speak on this platform about all things SARS-CoV-2 related. And I think Dr. Dorman did a really excellent job setting us up to understand some of the immune concepts that I'll discuss in the next 15 minutes or so. So what I wanted to talk about today is how vaccines work, okay? How vaccines work at the level of the individual, but also at the level of the population. Obviously, this is something that we're concerned about with the ongoing SARS-CoV-2 pandemic, and we're seeing transmission events increase every day. And we want to know when the end is near, when we can stem the spread of this virus and go back to our normal lives. So I'm going to talk about how vaccines work at the individual level, at the level of the population, and then we can tie that into what a SARS-CoV-2 vaccine might look like, what's coming in the future. And I can tell you that there are a lot of vaccines in development, a lot of vaccines that are in the pipeline. Okay, so how does a vaccine work? All right. The easiest way to think of a vaccine is that it's a rehearsal or a practice for your immune system. Right? We give you either a piece of the pathogen, sometimes in some cases, the whole pathogen. And what that does is it prepares your immune system if it were to see that pathogen later in your lifetime. Okay, it primes your immune system so that the next time you see it, you're able to fend that pathogen off, hopefully without ever being infected. Okay, that's the overarching goal of a vaccine, is to prevent infection, full stop. Now, vaccines take advantage of some key properties of our immune system, right? Namely, that our immune system has memory. It can actually remember a pathogen that it's seen before. Another key property that vaccines take advantage of is that our immune response actually gets better over time. So what does that gets better mean? I want to unpack that a little more. Well, it's actually the case that upon subsequent exposures, when our immune system sees a pathogen again and again, the quality of the immune response improves, and also the magnitude of the immune response improves, which means the first time you see a pathogen, you will elicit an immune response, but it's going to take some time. And it'll be a good response, but it's not a great response. Your immune system is able to store some of the information it gets from the pathogen away. It improves itself. And then when you see that pathogen again, you respond faster. The magnitude is higher, so you actually have a greater immune response, and the quality is better. So instead of a good response, you have a great response, right? And that is really what vaccines are taking advantage of, okay? That the second time you see a pathogen, you're going to respond in a far more robust fashion. Okay, so keeping that in mind, I think what we can build on there is talking about what Dr. Dorman told us in regards to the virological aspects of SARS-CoV-2, okay? Namely that we have this spike protein that is required for a virus to gain entry into our cells. And the key point there is that viruses need to access our cells in order to replicate, in order to make lots and lots of copies of themselves. If they can't get in, they can't replicate, they can't make copies, not only do you not become infected, you also don't go on to transmit that virus to other individuals. So most of the vaccines that are in development for SARS-CoV-2, the goal of these vaccines is to elicit antibodies to that spike protein. Okay, so antibodies are just molecules produced by our immune system. They're just proteins produced by our immune system that can help by binding a pathogen. The idea here is that if your immune system was primed, it had had this rehearsal against SARS-CoV-2, these antibodies would already be present. And if you were to be exposed to SARS-CoV-2, those antibodies could bind to the spike protein and prevent entry into the cells. Okay, and that's what we refer to as sterilizing immunity, because it means that your cells aren't even infected in the first place. There are other ways that antibodies can work, but this is the easiest way to think about the neutralizing capacity of antibodies. They block entry into the cell, full stop. Okay, so that's how a vaccine would work at the level of an individual. We would give you some piece of the pathogen, maybe an inactivated version of the pathogen, maybe an attenuated version of the pathogen. Your body would produce antibodies to that spike protein. And then if you were to be exposed to the pathogen in real life, you wouldn't even be infected because those antibodies would be primed and ready to go and prevent that infection from establishing in the first place. Now, how do vaccines work at the level of the population, right? It's easy to just think about us as one person, but obviously there is a global pandemic that's happening, and we need to talk about how we stem transmission at the level of the community, right? Now, vaccines at the level of the community rely on a property that's known as herd immunity. Okay? And what I mean by herd immunity, it's the maintenance of a critical level of immunity in the population that will therefore stem the transmission of the virus. Okay, and the easiest way to think about this is that if you have people that are immune in the population, and they're immune because they've been vaccinated, they can no longer be infected. Therefore, the virus can't infect them and they can't go on to transmit to others. And so I'd like to show an animation here that helps to illustrate this concept of herd immunity. And I think given our audience here that we're talking about a workplace environment and safety in the workplace, you can think about this in terms of a workplace exposure. Okay, so what you're seeing here with this graphic, and it will continue to play over and over, so we can follow along as the animation continues to play. So what you're seeing here is a population, and in that population, let's say in that workplace, there are people that are susceptible, and those are shown in blue, and then there are people that are immune, and those are shown in yellow. And what you're seeing here as we move across the animation, working from left to right and then down into the second panel, is that the number of immune individuals in this workplace environment is increasing. So we're moving from no immune individuals all the way up to 95%. Right? What you can see then is that as the number of immune individuals in the population increases, the transmission of the virus is stemmed. We essentially stop the spread of the virus because those individuals can no longer be infected and can no longer go on to transmit to other people. Okay, so, and you can see that actually the percent of people that need to be immune in order for this to work needs to be quite high. Okay, it's not, it differs for different viruses. So if we talk about what percentage of people need to be immune in order to achieve herd immunity for, let's say, measles or mumps or influenza, it differs depending on the virus, but it's usually somewhere in the high 80s or 90% of people that need to be immune. Okay, so again, if we take this back to an example in the workplace, if you were to experience an exposure in the workplace and no one were to be protected, no one had any immunity, you would expect that the virus would spread very rapidly in that environment. However, as we add immunity into the population, we remove susceptible individuals, we can see very nicely that that stems the transmission. And this is what we're working towards in developing SARS-CoV-2 vaccine candidates. We want to be able to provide immunity to the population, and that ultimately is going to stem the spread of transmission. All right. Okay. So keeping in mind how vaccines work at a population level, I'm just going to transition to talk maybe in the last 5 minutes or so about what vaccine candidates for SARS-CoV-2 could look like. Now, as I mentioned before, there are a lot of players in the field. The WHO has listed over 100 vaccine candidates that are in some phase of clinical trials. 42 of those candidates are actually in human trials, with 10 candidates being in phase 3 clinical trials. And phase 3 is the most advanced stage of testing. It actually looks at the efficacy of the vaccine. So we already have 10 vaccine candidates that are already in phase 3 trials, which is reassuring from a public health standpoint, but we're still a ways off from having those be available to the general public. Okay. There's been a lot of questions recently about how a SARS-CoV-2 vaccine candidate could work, right? How long the protection that it elicits would last for, all right? And how often you might have to be revaccinated, and whether or not it's a concern that the virus might change over time, and then that could affect the vaccine's efficacy. What we know about coronaviruses from studying other coronaviruses, like the seasonal coronaviruses that cause common colds, is that our immunity, our natural immunity that you would experience through infection, if you were to be infected with a seasonal coronavirus, it does wane over time. Okay, and that is actually normal for the immune response. There's been a lot of discussions, I think, recently about how the immune response to coronaviruses wanes over time, which are important discussions, but it's also important to note that all immune responses wane over time. So we need to ask whether or not that's significant in this case. But going back to our conversation about seasonal coronaviruses, what we know from studying those viruses is that immunity seems to last for a few years. Okay, so if we think about a SARS-CoV-2 vaccine candidate, we probably won't be able to get one shot and be protected for our entire life. We do have some vaccines for viruses like that. All right, I'll point out that the measles vaccine, most people get 2 shots and they're protected. 97% of people are protected for the duration of their lifetime. Then on the other hand, that's one extreme. On the other extreme, we have our flu shots, okay? And it's flu season. Hopefully everyone's going to their pharmacist or doctor to get their flu shot. And we know that we have to get our flu shots every year. Okay, so we have some vaccines we can get once and be done. We have some vaccines that we have to get every year. It's likely that a SARS-CoV-2 vaccine would be somewhere in the middle here. It's probably a vaccine that we might need to get every few years, maybe every 2 to 3 or every 5 years. But that really shouldn't be an obstacle in long-term immunity. There are many vaccines that we have to get booster shots every so many years. Okay, so even if our immune response was to wane over time, that's not a major obstacle in the production of a vaccine. And maybe I'll just end here by having a note on vaccine safety, because I know that above all should be our primary concern in the development of a vaccine. Obviously, these vaccine trials are happening at an accelerated rate. Typically, the normal timeline for a vaccine to move from the lab into into the clinic and into eventually humans takes somewhere around 15 to 20 years, okay? And it happens in a stepwise fashion where we have our preclinical results, then that you move the vaccine into phase 1, phase 2, phase 3, and so on and so forth. And obviously there are licensing hurdles to overcome that are related to vaccine production and pharmaceutical industry. Our timeline has become shortened for this particular vaccine because of the urgent medical need. All right, but that doesn't mean that we're cutting corners safety-wise, okay? What has actually allowed for rapid development is the fact that we have other pathogens that have informed SARS-CoV-2 vaccine development. Namely, we had vaccine candidates for MERS and SARS-CoV-1, okay, the first SARS that circulated in the late 2000s, okay. Our information about those candidates and how they work and our safety data for some of those vaccine platforms has accelerated the development of SARS-CoV-2. The other thing to say is that because of the urgent global medical need, there's been a huge financial investment from all sorts of biotechnology companies. And you'll know that many of the big companies like AstraZeneca and Johnson & Johnson already have vaccine candidates in phase 3 clinical trials. So these companies are taking on a huge financial risk and starting the process of developing the vaccines even before they've been licensed. And that also helps to shorten the timeline of bringing the vaccine to market and to actually making it available to the public. That is all to say that we are still following all the normal safety protocols that are in place for these phase 3, phase 1, phase 2, phase 3 clinical trials. And we're not sacrificing safety for efficacy here. And the governing bodies like the FDA have been very clear that they will not cut corners in licensing a vaccine. It will still have to meet the criteria that they set out in order to be acceptable for the public.
Wow, thank you very much, Dr. Mullarkey. That was fantastic, and I even just from the get-go, I like the idea of vaccines as a rehearsal for infections. So, and yeah, we've got a whole bunch of questions that we can dig into. So this is great. But our first question is going to go to Dr. Dorman, and this touches on the angiotensin, where you mentioned how inflammation, how it plays a role in inflammation, and I know you're also you've done a lot of research on how diet plays a role in inflammation. So in susceptibility to, say, SARS-CoV-2, how much should we be considering diet, in addition to, yes, we've got the vaccine coming, but should we be priming ourselves in other ways that you can talk a little bit about?
Oh, that's a good question. I didn't see the conversation going in that direction. Yeah, angiotensin 2 is going to cause some direct inflammatory effects. And so an imbalance of it is partly through reactive oxygen species. So diet most easily comes into the scenario in two ways. Primarily because a healthy diet is associated with high levels of antioxidants. And antioxidants will damp down. So the diet's not having a direct effect on your angiotensin 2 activation, but it will have a protective effects on the downstream effects of that hormone, right? So having a steady stream of antioxidants will counter the inflammation and prevent the downstream damage, which would include primarily atherosclerosis, which is going to be linked to your cardiovascular type diseases and sort of the metabolic syndrome effect associated with type 2 diabetes. But I also like to highlight with diet that healthy diets have a lot of the essential oils, which can also protect the damage of the cells themselves. So, chronic inflammation can kill a cell or damage parts of the cell. Your essential oils will protect that. And the essential oils, as well as some of our essential vitamins and minerals, are critical to supporting the immune system too, right? So we talked about that importance of having a robust immune response. And even the ACE2 protein has zinc in it, right? Your immune system relies on adequate doses of zinc, of vitamin D. That's a popular one that people are currently talking about, and all of the other essential nutrients. And although most Canadians are not deficient in many of these nutrients, many Canadians are insufficient, and so they're just not as well protected. So diet isn't going to cure you, but it's going to protect you. I call it your personal protective equipment. Make your cells as resistant as possible before you have the virus attack you.
Thanks, Dr. Dorman. That makes sense. It's not, you need to be a kind of holistic in the way that you kind of approach your personal wellness. And then, so Dr. Mullarkey, so actually, one thing that jumped out was just, I had no idea there's so many, like the number of vaccine candidates. That's huge. Now, do all like vaccines, I know you mentioned, so they've got that memory of your body for your immune system with the memory, and it gets better over time. Can you talk maybe a little bit about the different types of vaccines? Like how are there 42 different candidates, maybe like attenuated versus not, and how that plays a role?
Yeah, so there are lots of different types of vaccines, and there's some traditional methods that we have relied on for a very long time. And then what we're also seeing is newer developments, newer platforms that can be used for vaccine delivery. So some of the oldest ways to make a vaccine are to actually just to inactivate the pathogen, to kill it, either through heat or through chemicals. And then the pathogen is dead, but it still has all of the antigens that exist when it's alive and it's actually replicating. So when you give a dead pathogen to the immune system, it can still see that spike protein, for instance, and generate an immune response. But importantly, vaccines are safe. They don't cause disease, right? And that's easy to envision with a dead pathogen. Sometimes we can give not just a dead pathogen, we can give just pieces of the pathogen. And in that way, we generate a very specific immune response. Another way that we can make a vaccine is by attenuating a pathogen, which means that it is able to replicate, but it is only able to do so at a very minor extent. Okay, so it can replicate a little bit, but not a lot. And we make that by actually deleting genes from the viral genome that prevent it from being able to counter the host immune system to actually complete its infectious cycle. So some of the vaccine platforms that are in development do exactly those things. They're using attenuated pathogens, they're using chemically inactivated pathogens, they're using just pieces of the pathogens that are produced. Some of the newer vaccine platforms are things like viral vectored vaccines or RNA vaccines. And those are really two of the leading candidates that we're seeing. So the vaccine at Oxford, out of the Jenner Institute at Oxford, is a viral vectored vaccine. What that means is that we actually use a different virus to deliver the spike protein as an antigen. Okay, so we use a virus that is not replication competent. It can't actually infect the cell, but it can't actually replicate. It can infect the cell and it can deliver this antigen. So it's a kind of a safe surrogate of the coronavirus vaccine. So that's the vaccine that's being developed. It's in phase 3 trials with AstraZeneca. The other vaccine candidate that is in phase 3 testing, and this is coming out of a company called Moderna, and they are developing this along with the Vaccine Institute in Washington, D.C., the Vaccine Research Institute. It's an RNA-based vaccine. Okay, and that's not actually a platform that we've seen before. It is literally just RNA. It's just nucleic acid that's delivered into the cell. That RNA, the cell can take that RNA and then it can translate it into protein. So it takes the genetic material, it delivers it to a cell, and then the cell makes the protein itself. So it would take the RNA, it would make this spike protein itself, and then it could generate an immune response to that. So that's a really new technology that hasn't been licensed before. The preclinical results are pretty exciting. It seems to generate robust immunogenicity in phase 1 and phase 2, and there seem to be fewer side effects, probably as compared to some of the other vaccine platforms.
Wow, that's I hadn't heard of that before. So that's pretty cutting edge stuff. That's exciting.
Right, and it very exciting. There are some questions related because it's not a licensed vaccine platform that we've used before. There are some questions around development and manufacturing, in particular, manufacturing and scale. Okay, so if we think about this is a global pandemic, how many billions of doses do we need to stem SARS-CoV-2 transmission? Because the whole world, mostly besides people who have already been infected, are susceptible. So it could take somewhere around 16 billion doses of vaccine in order to stem the spread of transmission. And being able to scale up these platforms to deliver those doses could present some challenges that we haven't experienced before. And it could also delay the delivery of a vaccine to market.
Right. Thank you. That is a very good important. Just when we're talking global, that is a whole bunch of other factors that pop up that we haven't seen before. So that's a really interesting point. And we do have another quick question for you, Dr. Mullarkey, from the audience. And then in terms of, say, how if one of these candidates vaccines is successful, would it protect us from future SARS coronaviruses like MERS, or is that something we could expect?
Yeah, I mean, it kind of depends. It depends how similar this new virus is to SARS-CoV-2. So what we know from other coronaviruses like MERS and SARS-CoV-2 is that vaccines developed against those particular pathogens, they confer partial protection. Okay, so we can see what is called a cross-neutralizing antibody response. So your immune system can make antibodies to MERS and SARS-CoV-1. There are some similar features to SARS-CoV-2, and that provides partial protection. So it would depend on how similar or different the pathogen was to the SARS-CoV-2 strain that's circulating now. If it was very similar, it might provide a high level of protection. If it was very different, it might provide very little protection.
That makes sense. And so it's only, it's just a matter of time. We'd have to wait and see and what nature.
Yeah, and these zoonotic infections, right? All the evidence points to the fact that this virus probably originated in bats. They can be hard to predict.
Yes, absolutely. Thank you. And then, so I think we've got time for one more question. And so I'll put it back to Dr. Dorman. And so kind of considering everything we've talked about here, is there something that, a takeaway for workplaces when it comes to vaccines and return to work that you could talk a little bit about, Dr. Dorman?
Well, I mean, building off of what Dr. Mullarkey said, I think what's partly exciting is that this rapid investment and broad-scale innovation is going to have long-term consequences for the prevention of future disease, including diseases that may be like SARS or MERS that come up, but maybe even more broadly. Like this is really, I think, unheard of, the scale with which globally companies have responded and developed this. And I mean, I think from an innovative point of view, I think we're seeing that across the board in occupational settings. Everybody has been innovative, and I think we're going to see a lot of long-term positive outcomes because of that will be meaningful. I know that's hard to sort of focus on when we're in the midst of the awful, but I do think that positive outcomes will come from that. I guess being a health promoter, I think it also speaks to me about the importance of daily practices of maintaining our general health at all ages, right? We are a population of aging individuals, and I think maintaining that constant baseline health is important at every stage of our life. And so what I try to encourage people, Dr. Peguerero talked about this last week. Crises is a moment for change. It's an opportunity for change. Take the opportunity today to induce personal change, whether that is going for a walk every day, or meditating, as Dr. Kahalik recommended, or making some dietary changes to include more plants so that you're getting a healthier diet. Now's the time, because when there's all kinds of things happening, your behavior patterns change, and you can instigate new positive outlook. So that would be my final message: is grab the opportunity today, because it presents itself.
Thank you very much, Dr. Dorman, and really, so thank you very much to Dr. Sandra Dorman and to Dr. Caitlin Malarkey for taking the time today to share your expertise in such an accessible manner. You've really helped, like me for one, understand kind of how the biological infection, the mechanisms behind it, and why vaccines work and why they're important. So thank you very much to both of you today. And at this time, I'd like to thank all our previous panelists as well, and all the CROSH personnel who contributed to making the CROSH COVID Conversations a successful series. And thank you to everyone in the audience who took the time to tune in and submit your questions. So while CROSH COVID Conversations may be over, if you want, you can watch the whole series at any time at CROSH.ca. So once again, thank you, Dr. Dorman, thank you, Dr. Mullarkey, and from all of us at CROSH, wish you stay well and stay safe.